Welcome to our webinar hosted by Quality Assistance. I'm Julie Gia, Corporate Communications Manager, and I will be your moderator today. First of all, let's start with some practical points. The talk, presented by Arnaud Delobel, R&D Director at Quality Assistance, is entitled Analytical Workflows for Charge Variant Analysis of Monoclonal Antibodies. At any time during this 45-minute webinar, you will have the opportunity to ask questions through the Q&A dialog box of the webinar interface. I will be collecting them and Arnaud will be addressing them at the end of his talk. Let me now present today's speaker. Arnaud Delobel received his PhD degree in mass spectrometry from the Natural Product Chemistry Institute, ICSN, in France in 2004. He then further specialized in proteomics during his postdoc in the Mass Spectrometry Center of the University of Liège in Belgium. In 2006, he joined Quality Assistance and has held various management positions within the Operational Labs Department and the R&D Department, specializing in physicochemical analysis and characterization of biopharmaceuticals. He is since 2016 R&D Director and has just been appointed as an expert to the MAB Working Party of the European Pharmacopoeia. Arnaud, the stage is yours.
Thank you, Julie, and thank you all for being present today for our last webinar of the year. And the topic of the webinar today will be analytical workflows for charge variant analysis of monoclonal antibodies. So first, a very quick word about Quality Assistance. So Quality Assistance is a CRO based in Belgium, providing analytical services to the pharma and biopharma industry. We have more than 35 years of experience, and last year, for example, we have worked on close to 200 projects for 65 worldwide R&D companies. We are today 204 employees, and we all work on the same site in Donstienne, Belgium, in the south of Belgium. We offer product-dedicated support and customized project management, and everything we do is compliant with EMA and FDA regulations according to GLP and GMP environment. So here is the agenda for today. So first, I will talk about charge variants and what they are and why we should analyze them. Then I will present the different techniques that can be used for charge variant analysis, so icIEF, ion exchange chromatography, and I will do a comparison between the techniques. And finally, I will talk about the identification of those charge variants that can be done using 2D LC-MS or MS-compatible ion exchange chromatography. So first of all, what are charge variants? So proteins are made of amino acids, some of which can be acidic or basic. And depending on their pH, they can be either negatively charged, neutral, or positively charged. And how does that translate at the protein level? So at low pH, as you can see here, most of the amino acids will be positively charged, and the net charge of the protein will be positive. And the more you increase the pH, and the more you will have negative charge on your proteins. And at the end, a very high pH, your protein will have an overall negative charge. And that's when we define the isoelectric point, the PI, which is the pH at which the protein charges are balanced and the net charge is equal to 0. So that's what's shown here at the middle of the graph. You can see that there are as many negative as positive charge. So that means that there are still charges on the protein, but they are balanced and the overall charge is null. When we have a look to monoclonal antibodies, so this figure is taken from a publication in 2017 in Journal of Chromatography B, and you can see that for those 23 therapeutic monoclonal antibodies, the majority of them have isoelectric points that are above 7.0, and most of them even be above 7.5. So that means that at neutral pH, most of them will be positively charged. So let's talk now about the heterogeneity of monoclonal antibodies. So you all know that monoclonal antibodies are very heterogeneous due to many different modifications, and some of these modifications may have an impact on the charge of your protein. So first, methionine oxidation has a small impact on the charge, and you will observe these variants as basic variants. Pyroglutamate formation is also a modification that is common on monoclonal antibodies, but this one will have a moderate impact on the charge of your antibody. At the opposite, this modification, the C-terminal lysine truncation on the heavy chain, will have a strong impact on the charge of the protein, because if you have one or two lysines, you will have some basic variants that you will observe either by CIEF or by ion exchange chromatography. So this variant is something that is present at the beginning, so due to the production of the antibody, but is not something that will evolve during the life of your product. And glycosylation can also have an impact on the charge, because if you have some sialic acids, you will observe some acidic variants. At the opposite, if you have some non-glycosylated species, they will appear as basic variants on your chromatogram, on your electropherogram. And finally, a major degradation product on monoclonal antibodies is asparagine deamidation or aspartic acid isomerization. The isomerization of aspartic acid will have no impact on the charge, but the asparagine deamidation will have an impact, as I will show just now. So what is asparagine deamidation? So this occurs, for example, on this kind of structure. So you have here an asparagine with in red the side chain. It can cyclize, so you have these succinimide intermediates that can then hydrolyze either as an aspartic acid or as an isoaspartic acid. So you have here an asparagine residue that converts into aspartic acid or isoaspartic acid. So you will generate an acidic variant, and that happens during the life of your product. This mechanism is favored at higher pH, and the rate will be higher on asparagine residues that are more easily accessible to solvents. So, for example, an asparagine that is followed by a glycine residue. So this table is taken from a publication from Kali and al. In 2010, and it shows the major degradation pathways that you can have on monoclonal antibodies and the species that are generated. So I will not go through the table in detail, but you see that some modification can lead to acidic residues, or some other modification can lead to basic variants. And all these can be seen by icIEF or by ion exchange chromatography. And all these variants are considered as charge variants. So let's move now to the techniques that can be used to study charge variants. And the first one is capillary isoelectric focusing. So isoelectric focusing is something that is not really new. And the concept that you can do that either in a gel or in a capillary, but the concept is exactly the same. So you will work in a gel in which you have a pH gradient that is established before you load your sample. So that means that in your gel, when you go from the left to the right, you will have an increase in pH. So you have a pH gradient. When you load your sample, depending on the charge of the protein at the pH on the gel, it can be either positively or negatively charged, and you will apply a voltage. So proteins that are positively charged will move towards the negative electrode, and the opposite for the proteins that are negatively charged. And when they reach the zone for which the pH is equal to their isoelectric point, they are no longer charged. The net charge is zero, and then they will stop and they will be focused. So that is the main workflow that is used for isoelectric focusing. So as I said, this can be done either in gel, so that's the old way of performing isoelectric focusing. It can be a capillary IEF, but also image capillary IEF. So in a gel, the problem is that the throughput is quite low and the separation time is quite high, so it can take several hours. The resolution is not bad, but you have this drawback in terms of throughput. When you switch to capillary IEF, and that's what we can do, for example, at Quality Assistance on our PA 800 plus systems, you have an improvement, a clear improvement in terms of automation and in terms of throughput. And the separation time is also much quicker. You don't have a very high increase in terms of resolution, but the fact that you are able to automate the separation, the analysis, and that you decrease the separation time is a great improvement when you do that in a GMP environment. And more recently, we have the image CIEF that has appeared. So we do that either on icE3 or a Maurice system from Protein Simple. So you still have an increase in terms of separation time compared to CIEF. You have an increase in throughput. And at that time, you have also an increase in terms of resolution. So why do you have this increase in resolution between CIEF and imaged CIEF? So the separation principle is the same. So you have a pH gradient in your gel, and you perform the separation as I have described just before with a voltage that is applied on your capillary. When you perform only CIEF, in order to detect your sample, you have a mobilization step. That means that you will apply a pressure on your capillary and send the sample towards the detector. When you do image CIEF, you focus your sample and you have a camera, UV camera, that is able to perform direct whole column detection. That means that you will take a picture of your capillary to have a view of your separation. So you avoid this mobilization step, and then you are able to improve the resolution. So here is an example of separation that is done by icIEF, for example, here on Adalimumab. In the x-axis, you see it's not really minutes, but it's actually the PI. So you are able to measure the PI of your sample. So here about 8.8. And you can see the acidic variance here and the basic variance. So you see that we have a good separation between the different species. The basic variance here most probably correspond to lysine variance. So one lysine and two lysines on the heavy chains. And the acidic variance most probably correspond to deamidated species. The advantage of this technique is that you use a kit. It does not involve a lot of method development, and you can use a generic method that is able to separate most monoclonal antibodies. And that's what is shown here on this slide. You have the separation for rituximab, trastuzumab, and adalimumab using exactly the same method. And you see that for monoclonal antibodies with different PI, we have a good separation, good resolution, with exactly the same method. So in icIEF, you can obtain good separations quite easily for most antibodies. The method optimization is very quick, and you can use a systematic approach for this optimization. The only drawback is that the sensitivity may be limited for minor isoforms due to the signal-to-noise ratio that is not really high. This method can also be used for antibody-drug conjugates, and I have shown here the example of Kadcyla, trastuzumab emtansine, which is a lysine-linked ADC. So you have here the naked antibody, and each peak corresponds to one drug that is linked to your antibody. So one drug, two drugs, three drugs, and so on. And why is that? Is that because when you have a drug that is linked to a lysine, you have a basic site that is removed from the antibody, and you generate acidic species. So let's move now to ion exchange chromatography. So here is the principle of ion exchange chromatography. So as I said, most monoclonal antibodies have a pI that is higher than 7. So we will work in cation exchange mode. We will work at a starting pH that is below the isoelectric point to be sure that the mAb is positively charged. So as it is positively charged, it will bind to the negative charges that are present of the surface of your cation exchange column. The samples will be more retained if they have more positive charges, and the elution will be performed either by increasing the salt concentration, and in that case, there will be a competition between the cations in your mobile phase and the protein that is bound to the column, or by increasing the pH. If you increase the pH, you will lower the number of charges on your protein, and you will finally reach pH that corresponds to the isoelectric point of your protein, and these proteins will be removed from the column and they will elute to the detector. And finally, using this mode of separation, acidic species will be eluted first and then the basic species. So why would you like to use salt gradients versus pH gradients? So in salt gradients, as I said, you will increase the salt concentration to elute the protein from the column. Most of the time, you'll need a product-specific method development. You cannot use generic methods for salt gradient separations, especially if you want to have an optimum resolution. You will also need to prepare your mobile phases manually, so it's quite time-consuming and it can be a source of error in the lab. But using salt gradients, you have the possibility to get very high resolution if you optimize your method, and it is independent of a commercial kit. If you use pH gradients, you will increase the pH to elute the protein from the column, and in that case, you have generic methods that can be achievable using this approach. In most cases, the resolution will be lower compared to salt gradients, as I will show later. And if you want to have a method that works really well, you need linear gradients to get correct and robust separations that will be applicable to all monoclonal antibodies. Also, in order to get linear gradients, it's quite complicated to do this by hand, and there is the possibility to use commercial kits, as I will show later. So here is an example of the separation of Adalimumab using a salt gradient. So it is a method that was optimized in our labs using a thermomap SCX10 column, a MOPS buffer at 100 millimolars at pH 7.1, so below the isoelectric point of your antibody that is close to 9. And we use a sodium chloride gradient between 5 and 95 millimolars. And you can see here that we get a nice separation between the different acidic variants and the different basic variants. And we get something that is quite similar to what we obtain with CIEF. But I have to mention that this method development was quite complex in order to optimize both the nature of the buffer, the molarity of the buffer, the starting pH, and also the gradient of sodium chloride in order to elute the protein. So if you want to have a method development that is much simpler, in that case, you can use pH gradients. And as I explained earlier, doing pH gradients by hand is not something that is quite easy, especially if you want to get a linear pH gradient. So you can use different commercial solutions that were commercialized. So Thermo Scientific has commercialized pH buffers that can be used with their columns, but also with columns from other vendors that give nice results. And more recently, Waters has developed a BioResolve column and also quite different approach because they developed both the buffers, but also the column that is optimized to be used with those buffers, which allows to get very nice resolution with this kind of columns and with this kind of buffers. So here is the example of what we can get with the Waters solution on Adalimumab. You see that you get a nice separation between the basic variants, and you observe those acidic variants in the other way. So you get a separation that is not as good as what we obtained with the salt gradient method that was specifically optimized for adalimumab, but you get anyway a separation that is acceptable. But the main advantage of this solution is that you can use it for most monoclonal antibodies and have a platform method. And that's what I show here. So for rituximab, trastuzumab, and adalimumab, using exactly the same method with the conditions that are recommended by the supplier, you get correct separation, a good sensitivity, and you have the possibility to use the same method without any optimization for different monoclonal antibodies. If you want to optimize the method, you can still play on the gradient that you use, the slope, the starting and end pHs. And in that case, you can slightly improve the separation, but in our hands, we were not able to obtain separation that were as good as what we obtained with salt gradient, specifically for adalimumab. If you want to compare also the two commercial solutions, we have used the thermo CX buffers on the thermo columns, the waters buffers on the BioResolve waters column, and you can see the separation that we get with the conditions that are recommended by the supplier without further optimization for adalimumab. And what you can see here is that with the water solution, we get a better resolution for both the basic and acidic variants. And even if it's still acceptable for thermo, the resolution is lower. We have not tried to improve the separation, but probably you could improve the separation both for thermo and water solution, but it will have to be product specific, and so you will have to perform method optimization for each monoclonal antibody that you would like to analyze. So a question, when you have all those techniques, icIEF, salt gradient IEX, or pH gradient IEX, which one should you choose when you have to analyze your monoclonal antibody? So I have compared here the analysis of adalimumab with the three methods that I have shown before, the pH gradient, icIEF, and salt gradient. So you see that pH gradient is the method that probably leads to the lower resolution, and icIEF and salt gradients give comparable resolution. You should not focus on the relative intensities of the different peaks because the analyses were done at different moments and with different batches of adalimumab, but you should focus more on the resolution. And you can see that resolution is quite comparable between icIEF and salt gradient. You may have some improvements for some variants and lower resolution for others, but globally, it's quite similar. So which method should you use for which application? icIEF is a generic method with little or no optimization. You obtain a good resolution that may be product dependent, but you may have a limited sensitivity for minor isoforms. pH gradient IEX is also a generic method with little or no optimization. You will have a limited resolution that may be product dependent, but you have a very good sensitivity. And finally, salt gradient is a mix of both methods. You will have to perform a product-specific development, but you get the optimum resolution and a good sensitivity. So depending on your application, on the stage of development of your product, you may want to choose either of these techniques. So we have seen how we can separate the charge variants. Let's move now to the identification of those variants. So how to identify charge variants observed either by ion exchange chromatography or capillary IEF? So the identification of the variants is something that is required by the guidelines as part of product characterization. So of course you will not do that for all the batches and as a routine test, but you need somewhere in the development of your product to perform the analysis and the identification of those variants. icIEF cannot be coupled directly to mass spectrometry, so it involves very low volumes. It does not use MS-compatible buffers, and there's also a technical complexity for the coupling of the capillary isoelectrofocusing to the electrospray mass spectrometry. So I must say anyway that there are some commercial icIEF equipments that are now available, but we are not equipped with those systems, and also it's not something that can be routinely used. So you may not be able to use exactly the same system for routine testing and for characterization, and so you may have some differences in your separations. If we have a look to ion exchange chromatography, the main drawback is that mobile phases are not MS compatible due to the salt content that you have in your mobile phases. So how can you identify those variants with the limitations that I have described here? So the first way to identify variants is to use carboxypeptidase B. So it will be used only to identify lysine variants, but it's anyway something that can be done quite quickly. So carboxypeptidase B is an enzyme that cleaves the C-terminal lysines on your heavy chains. So here you see adalimumab analyzed without digestion. And if you digest your sample with carboxypeptidase, you see that some basic variants, so the two that are shown here, have an intensity that is significantly decreased after digestion. That means that those variants correspond to lysine variants on your antibody. At the opposite, this variant here is not significantly impacted by the digestion, so most probably does not correspond to a lysine variant. And your acidic variants are not impacted either by the digestion. So that can be quite useful to determine which basic variants are lysine variants, but it does not allow you to identify other variants. So you have to use other techniques, and 2D LCMS is one of the techniques that can be used to do that. So at Quality Assistance, we're equipped with this system, which consists in a Xevo J2XS QTOF coupled to 2D UPLC with an H-class bio as the first dimension and an I-class system for the second dimension. Everything is controlled within Unify, so this ensures full GMP compliance. And we use this system in a multiple Heartcut mode, and I will describe now what it corresponds to. So what is multiple Heartcut to DLC? To do multiple Heartcut to DLC, you need first to acquire your first dimension chromatogram and determine which are the fractions that you would like to analyze in your second dimension. So here, for example, you will identify two fractions that you would like to send to the second dimension to be analyzed by LCMS. Then, during the first step, you will have this first part of the chromatogram that does not correspond to something that you want to analyze in the second dimension. So the flow will go through the first dimension column and be sent directly to your first dimension detector, which is a UV or PDA detector. Then you will arrive to your first Heartcut here, and you will switch your valve in order to send the flow of your first dimension column to a trap column, which is a reverse phase column, a short reverse phase column that will be used to trap your sample. In ion exchange chromatography, you use only aqueous buffer, so they will be stacked at the entry of your reverse phase column. Then you have the third part of your chromatogram, which corresponds to fractions that are not of interest. So you will switch your valve and send everything to the UV detector. And you will reach your second fraction of interest, your second Heartcut. You will switch your valve once again, and you will send the fraction to the second trap column. So in our system, we are able to trap up to 7 fractions, and to perform with only one run of IEX, trap 7 fractions to send to 7 trap columns. Then you have a step of desalting sample. So you have here an additional pump that is used to send either aqueous buffer to your trap column to desalt your sample, or if you want, you can send other types of mobile phases, and we use it also to send DTT on the column to perform online reduction of your samples. And finally, the last step is to analyze the different fraction in the second dimension. So we have a second pump, a BSM in that case. We backflush the trap columns sequentially. So first you will backflush the first trap column, send it to the second dimension column, which is a reverse phase material that is used for both desalting and separate the different species in the second dimension, and everything is sent to the QTOF. And once you have analyzed your first fraction, you do exactly the same with your second trap column to analyze your second fraction. So here is a case study on nivolumab by 2D LCMS. So the first dimension is a BioResolve SCX column, and we use a pH gradient with the commercial pH buffers from waters. The trap column is the BioResolve RP column, 3 centimeters column. The trap column is set as 80 degrees, and we perform before the second dimension a desalting step of 10 minutes with 0.1 formic acid in water. And finally, what is trapped on the column is separated by the second dimension column, once again, a BioResolve reverse phase column from waters with a 0.1% formic acid in water and 0.1% formic acid in acetonitrile. So here is the first dimension chromatogram that we obtain with the different acidic variant and basic variants and the main isoform. So this is the example for nivolumab. And that's the mass spectrum that we obtain for the main isoform. So you can see that we can easily see the different glycoforms on the antibody. The first one that contains G0F on both chains, G1F on one chain and G0F on the second heavy chain, and G1F on the two chains, or G1F and G0F and G2F. So we can easily identify the main isoform. If we have a look here to the first basic isoform, so it's quite low in terms of intensity, so the mass spectrum is not as good, but you see that we have a mass here that could correspond to non-glycosylated species, so the antibody without any glycan on either of the two heavy chains. The limitation here is that at these masses, the mass accuracy is not as good as what you can get with subunit analysis. And for small, for low amount isoforms, you may not get a very good sensitivity. The second way to perform the test is to use roughly the same analysis. So the gradients are slightly different for the trap and for the second dimension. But we have included a reduction step with 10 millimolar DTT on the trap column to reduce the sample in the light and heavy chains. And what you can see here is that on the second dimension for the main isoform, this time we detect two species, the light chain, on which we are able to identify very low variants. So here the pyroglutamic acid, and the heavy chain on which you can see the main glycoform, main glycoform G0F, G1F, and a very low amount of G2F. So using this way, this reduction, you have a gain in mass accuracy, in sensitivity, and you are able to see lower abundant isoforms. If we have a look now to the first basic isoform, you see that the mass spectra we obtained are much better compared to what we had for the intact mass. You see once again the light chain, and then you see also this species, a non-glycosylated heavy chain, together with a glycosylated heavy chain. So we probably have a mixture due to the fact that we do not have a full resolution between the two isoforms, and we have probably in the basic isoform a contamination by the main isoform that you see here. So that's a way of working. We do 2D LC-MS. It works well, but it's true that it takes some time and you need a 2D LC system to perform this kind of characterization. Another way of performing the analysis is to use ion exchange chromatography directly coupled to mass spectrometry. So MS-compatible ion exchange chromatography is described in the literature, but usually it does not give very good chromatographic performance, either if you use salt or pH gradient. So many academic groups have worked on MS-compatible mobile phases, but recently a commercial kit was also commercialized by Water, so the ion-enhanced CX system, using once again the same BioResolve SCX column. So here is the chromatogram we obtained by icIEF on nivolumab. So here is the UV trace. You see that we have something that is quite similar to what we obtained by SCX UV, so with the non-MS compatible mobile phases, but we have a slightly lower resolution. So it's a compromise to have. Either you have the MS compatibility or you have an optimum resolution. But the main advantage is that it can be directly coupled to mass spectrometry. So here, that's what the mass spectrum that we obtained for the main isoform of nivolumab on the BioAccord system. So the BioAccord system is the routine high-resolution mass spectrometer that can be used very easily, even by people that are not experts in mass spectrometry. And you see here that you get a very nice mass spectrum and a very nice deconvolution, even better compared to what we had by 2D LC-MS on a Xevo J2XS QTOF. What you can see here, if you are familiar with MS, is that we observe the charge envelope at much higher M/Z ratios. So we are here between 5,000 and 7,000, and that is explained because we have to work in native mass spectrometry, because of course the mobile phases by ion exchange chromatography are not denaturant, and so you observe the native species, and that's why you need to go high in terms of M/Z ratios. But anyway, even on a routine mass spectrometer, you get very nice spectrum with very nice deconvolution. If we analyze the minor species, you see here that we have peak 3 and peak 4. For the peak 3, we detect the monoclonal antibody with one non-glycosylated heavy chain. So we still see G0F and G1F. And for the peak 4, we get the mAb with two non-glycosylated heavy chains. So here, without any glycan on the chain. So that's something that we were not able to see easily by 2D LCMS because of the resolution of the separation, but also to the fact that we have to do a Heartcut with the minimum amount of time, and we may not have the full resolution to detect all the species, and we have some coelutions in our second dimension. So a few take-home messages now. So I have shown several techniques for charge variance analysis. So all those techniques are available at Quality Assistance. You can use icIEF, so we do that on icE3 from Protein Simple, or the Maurice system also from Protein Simple. This technique is very easy to implement. It's based on kits, and even easier on the Maurice system. You get a very good resolution. It can be used as a platform method with no optimization, or with minimal optimization if you want to have a product-specific method. And the only drawback is that the sensitivity may be quite limited for for minor isoforms. You can also use pH gradient, IEX. It's easy to implement. Once again, it's based on kits. It can be a platform method. You have a limited resolution that can be slightly improved if you optimize your method for your specific product, but you have a very good sensitivity, and you now have the possibility to use MS-compatible commercial solutions. And finally, salt gradient ion exchange chromatography. You will have to do a product-specific method development, but then you will obtain a very good resolution and a very good sensitivity. So most of the time, we use our H-class bio systems to perform this kind of separation, either for pH gradient or salt gradient. If you want to identify your charge variance, which is required by the guidance, you will have a lot of difficulties to do that after icIEF, even if there are now some commercial systems that can do that. But the variance you observe by ion exchange chromatography can be identified by 2D LCMS. If you want to use MS-compatible ion exchange chromatography, that's an innovative approach that now allows the direct identification of the variance, even for minor variants. And I have not talked about that today, but all that is presented here is done at the intact level, or even at the subunit level if you do 2D LCMS after reduction. But if you want more information, you can do a peptide mapping by LCMS using a multiple attribute monitoring approach. And if you use a suitable digestion with trypsin in optimized condition to avoid the formation of deamidation and oxidation during the trypsin digestion, then you can determine which sites are deamidated and the amount of deamidation, for example, on each site of your protein. Finally, I want to thank my colleagues that worked on this project. So Camille, Claire, Anissé, Alicia, and Vanessa. I thank also the Région Wallonne for funding. And I thank you for your attention. I will be happy to take any question you may have.
Thank you, Arnaud, for your very clear and interesting presentation, and thank you all for your interest. Several questions were asked during this webinar. You can still send us your questions through the Q&A box. We are going to answer some of them now. Others will be answered later by email if we don't have enough time to cover everything. The first question we have, Arnaud, is the following. How do CIEF and ion exchange chromatography compare in terms of relative quantification of the variance?
Yeah, so it's true that it's not something that I've presented today. We do not have a lot of examples for which we have done both the CIEF and the ion exchange chromatography on exactly the same sample. Usually, you can get a good correlation between ion exchange chromatography and CIEF in terms of percentages of basic variance, acidic variance. But the main limitation, in my opinion, will be the resolution that you will get. You have seen that, for example, if you use pH gradient ion exchange chromatography, you may have sometimes a low resolution and acidic variance. And so when you will integrate your peaks, there will be a bias because you won't have the full resolution, and you will sometimes overestimate, sometimes underestimate the amount of acidic variance compared to icIEF. But when you use optimized conditions for which you get a very nice resolution both in icIEF and in ion exchange chromatography, so that's usually the case when you optimize your method as a salt gradient chromatography method, then you get a very good collaboration. So in that case, you will probably be able to compare the numbers that you obtain. One thing also that should be taken into account is that you measure your charge variance, but you do not work exactly the same way. So in icIEF, you take into account the total charge of your protein. In ion exchange chromatography, the charges that will be taken into account are those that are accessible to the solvent and then that can interact with the column. So charges that may be a little bit more buried into the structure of your protein will not interact with the column. So you may have some small differences.
Thank you. Another question. What is the product quantity needed for each method you presented?
Okay, so it depends on for icIEF, it's very low. So you will be more limited by the amount of sample that you need to put in your vial rather than the amount that is really used for the analysis. So it's very small. So icIEF can be used with very small amounts of sample. For ion exchange chromatography, it will of course depend on the column that you use. If you use some older columns, 4.6 millimeters, for example, and quite long, you may need to inject several, between 20 micrograms up to 50 micrograms of protein, depending on the complexity of your sample on the column. If you use some rather new columns, 2.1 millimeters, in that case, it will be a few micrograms that will be used. If you do MS, the amount of IEX-MS, for example, the amount of sample that you need to use in order to get a correct mass spectrum is quite low. But if you want to use UV, of course, you will have to use more. If you perform now for the identification for IEX-MS, once again, it's quite low. The example I have shown was done by injecting between 2 and 4 micrograms of nivolumab. If you do 2D LCMS, maybe you'll need a bit more of sample, but it's quite similar. But if you want to get a very good mass spectrum for minor isoforms, you may need to inject more. So it will depend on the application, on the type of column that you use, but usually between 2 and 10 micrograms of sample are enough, and I would say rather between 2 and 5.
Thank you. The next question is the following. CIF may be coupled to mass for characterization. Here, only 2D LCMS were presented. Is anything preventing from having CIF coupled with mass?
Yeah, so for the systems that we use, protein simple systems, it's complicated, and I would say almost impossible to couple CIEF with mass spectrometry. There are some challenges when you want to couple capillary electrophoresis with mass spectrometry. The first one is the low amount of sample that is used, the low flow rate that is used. and also the fact that if you want to couple something that is using a voltage difference to perform a separation, when you want to couple that to electrospray ionization, that can be some challenges to couple everything. So there are, with a regular protein simple system, I would say that it's almost impossible to perform MS coupling. So there are now some systems that exist commercially that are able to be coupled to mass spectrometry, and they are designed to be coupled to mass spectrometry. So you will have to use nanospray, nano electrospray instead of regular electrospray. So it's less robust in terms of mass spectrometry, but you will get nice results. Unfortunately, we are not equipped with this kind of system, like quality systems, at the moment. And also, I don't think that it's a system that could be used for routine analysis. You don't need MS to perform routine analysis. And these kind of systems are more dedicated to R&D work at the moment. So you would probably not be able to use the system that you commonly use for your routine test and that will be used for QC as the system that you will use for CEMS. And in that case, by using different systems of pharmalytes, electrolytes to perform the separation, you may have some differences in terms of resolution between your different systems. So it may be a challenge if you want to correctly identify your variants.
Thank you, Arnaud. The next question is, are there any trap columns available with immobilized enzymes like PNGase or trypsin?
So I'm not aware of immobilized columns with PNGAs, but there are some columns with immobilized trypsin. So the problem is that you could not perform the trapping on an immobilized trypsin column because we use the fact here that we use a reverse phase material to trap the sample, to also concentrate the sample and also desalt it. So as a trap column, it will be quite complicated to use an immobilized enzyme column. But it would be possible to use in the second dimension a column with an immobilized enzyme to perform some digestion and also analyze your sample and check which sites are deamidated or modified. At Quality Assistance, for the moment, we do not use these kind of columns. We have some applications that use IDS columns. So these are columns with commercialized by Genovis with so it's called Fabricator HPLC that we use as a first dimension, and then we use reverse phase as a second dimension to perform automated subunit analysis. If you want to see applications of trypsin digestion online with ion exchange chromatography, there are some examples that were published in the last months and last years by the team of David Guillaume in Geneva that performed a 3D and 4D LCMS. So you have some very nice examples of automated characterization of charge variants. So it's very nice and it works very well, but clearly it's not something that can be done in a regulated environment, in a GMP environment, because you need to modify a little bit your LC systems, but it's possible.
Thank you. Next question. Do you have an approach to compare icIEF and IEX results as they are considered orthogonal? Do you have a strategy to find the same components?
So we do not have a defined strategy, so it will be dependent on the product and on the separations you have. So yes, that's we usually consider that icIEF and ion exchange chromatography are orthogonal methods, and that's a way to identify the variants that you have in your CIEF if you do not have access to CIEF coupled to MS. In that case, what you need is first to have separations that are similar. So most probably you'll have to optimize your ion exchange chromatography method to get resolution that is similar to what you have in ion exchange chromatography. Then the approach I would have would be to isolate the different components that you observe by ion exchange chromatography. So collect your fractions and then re-inject them in your IEF method, cIEF method. So the opposite is not possible because it's almost impossible to collect sample after the cIEF separation and also to get enough material to be injected in the ion exchange chromatography. But that's what I would do. First, optimize your ion chromatography method to get a similar resolution to the icIEF. Then collect your different variants after the ion exchange chromatography and re-inject them by icIEF to compare the retention times and identify your variants and do the correlation between the two techniques.
Thank you. We'll be taking one last question now. Which method will you recommend for stability testing?
So we use both techniques for stability testing. We use both icIEF and ion exchange chromatography. It will depend on your sample, and it will also depend on the ion chromatography method, ion exchange chromatography method, and how it was optimized. If you use a platform method, so one of the methods I have shown using pH gradients, using commercial buffers, I would say that you may lack the resolution to see very small differences between your samples. So in that case, you would probably have to optimize a salt gradient method or to try to optimize your pH gradient method. But in our hands, we were not able to optimize very significantly the resolution by tweaking a little bit the gradient of the pH separation. So for a stability testing method, I would say either the icIEF method or a salt gradient product-specific ion exchange chromatography method. With one remark anyway, by icIEF, usually you get a lower sensitivity. So for the minor isoforms, it may be slightly lower. So if you have a lot of small isoforms, it may be tricky to get a very good limit of quantification. So in all the projects that we manage at the moment, some of them use icIEF as an identification test, but they use ion exchange chromatography for charge variant analysis. So it will be product dependent, but in my opinion, I would use probably, if I could choose and if I have time to develop a product specific method, I would use salt gradient ion exchange chromatography.
Thank you, Arnaud, and thank you all for your participation and interest. Please note that the recording of this webinar will be available shortly. Make sure you follow Quality Assistance on LinkedIn and Twitter to stay updated with our latest news. And of course, feel free to contact Arnaud for more information and if you have any other question. Goodbye, everybody.
Thank you. Goodbye.